Good. Good morning, everybody. Good morning to the second day of the virtual ILC Symposium. We'll start off this day with a discussion on how researchers, clinicians, and patient advocates can accelerate lobular breast cancer research. And I'm very delighted to introduce Leigh Pate, who will give the first presentation together with myself. I want to introduce Leigh, who has become not only a partner in crime, but also a dear friend of mine. So Leigh is a breast and a patient advocate. She is a Susan G. Komen AIS Steering Committee member and a Susan G. Komen Scholar and founder of the Lbca. I am a professor at the University of Pittsburgh. I co-lead the Cancer Biology Program and the Women's Cancer Research Center here. I'm also a Susan G. Komen Scholar and the founding chair of the Scientific Advisory Board of the Lobular Breast Cancer Alliance. Just a few words for the introduction. Leigh and I met at the 2016 Lobular Breast Cancer Symposium in Pittsburgh, and the symposium one was really very well received. And at the end, we got together and thought, okay, we should really do something for lobular advocacy. And we thought maybe the idea of really founding a advocacy group specifically for patients with ILC would be great. So we had a couple of calls, we met. We are very fortunate to be joined by a number of very motivated patients with ILC, which then really turned into ILC advocates. And that's how the Lbca took off. It really has grown over the years. We are very delighted that now other people have joined and stepped up. You will hear more later on from Laurie Hutchinson about it, but clearly it has grown. And the idea now is really, there is so much going on different continents to really discuss how not only nationally, but internationally, researchers, clinicians, and patient advocates can work together to accelerate lobular breast cancer research. With that introduction, I think I will give it over to Leigh Pate. Leigh, maybe you can take it from here.
Oh, thanks, Steffi. Hello, everybody. My name is Leigh Pate, and what we're talking about today is also a article that was just published in Cancers, and everybody, we're going to be joined by all the co-authors of that commentary for some Q&A and discussion at the end of that. So this is the article, and you can go check it out. It's fascinating reading. But what we wanted to start off with today was basically the presentation today is going to go over quickly kind of a historic view of lobular breast cancer and how that view is changing as we're learning more about biology and therapies, and how those might be differently applied to lobular. There's growing interest in ILC research, and that has corresponded with growing patient advocacy, which is really put a dynamic combination together that is with good opportunity to move research forward a lot more quickly. Then in this paper, we identified some challenges and some opportunities to work together to address some of these challenges to advance research. And then we'll discuss some ways that we can move forward. And then we've invited some of the organizations, the new advocacy organizations and groups to kind of briefly introduce themselves and talk about what they are working on, so that if anyone wants to learn more, get involved, that you know how to do that. And then we'll do a Q&A and discussion. So lobular breast cancer, if we can go back to the earlier slide, Steffi, thank you. It was first described in Foote and Stewart about 100 years ago. And while there was progress made in subsequent years, including defining some of the subtypes within ILC in the 1970s, the prevailing view was that ILC was basically a kind of another estrogen receptor positive breast cancer. And this view was shared by a lot of the eminent researchers at the time. And here's an example from the 1970s from physicians Edwin Fisher and Bernard Fisher, and they basically concluded that since the overall 5 and 10-year survival rates of patients with infiltrating lobular carcinoma are reputed to be similar, there are no special considerations regarding its management. They should be similarly treated. And they go on to say, whatever information is obtained in the next few years from ongoing clinical trials regarding management of breast cancer will be directly applicable to this variant. And I can speak for myself, as when I was diagnosed with early stage lobular breast cancer in 2011, this was certainly the view and how I, in my experience, working within the, trying to learn about the disease and getting care. So one of the reasons that may be contributing to this is that there seems to be a perception that lobular breast cancer is actually a rare disease. But actually, lobular breast cancer is quite common. As you know, breast cancer just became the most frequently diagnosed cancer in the world. And lobular is the second most common histological subtype. In the US, if you just pull out lobular breast cancers from ductal cancers and you rank them by prevalence in cancers of women, lobular breast cancer alone is the 6th most frequently diagnosed cancer of women. So this is a disease that hasn't received a lot of attention and is impacting a tremendous amount of women around the world. And Steffi, do you want to?
Yeah. So we just have 2 slides here on the biology. Quickly, we have heard a lot about this already yesterday and actually the day before, but just want to remind you that there are unique aspects of lobular cancer biology. We have heard yesterday that there are clearly a number of very unique variants within ILC. There are unique cellular structures and distinct clinical features, both in the primary as well in the metastatic setting. I think there's a unique metastatic pattern and presentation, in addition to some 2 metastatic sites which are shared with IDC. They are more frequently, they're almost all more 95% estrogen receptor positive or luminal A. There are the major hallmark of lobular is EKT mutations, and we'll hear more about this today. There are some other changes in genomic alterations, which are either different in the number or also in the location of the mutation. And, we really think that these changes in the molecular features, which again, I think we haven't even discovered all of them. There are other changes, let's say, DNA methylation and protein, and we really haven't looked at a lot of those, that they hopefully, and the clinical trials and data are coming out, that they provide potential for personalized therapy for ILC. There is some evidence already from clinical trials. We heard about this yesterday. There is some evidence that a response to endocrine therapy might be different. This is a tamoxifen. Dr. Metzger talked about this yesterday. There are clearly changes in imaging, and Dr. Linden talked about this yesterday. It's often occult on imaging. There are different responses to chemotherapy, higher rates of positive margins in surgeries. And we have had a lot of discussion. It started off actually with the keynote from Dr. Desmet that clearly there are challenges for the prognostic tests and assays, which weren't very often set up for lobular tumors. So clearly we need more research to understand lobular disease and to refine therapies. And Leigh, I'll give it back to you.
Okay, thank you. So Steffi already alluded to the first International Lobular Breast Cancer Symposium, which was held in 2016 in Pittsburgh and hosted by UPMC Hillman Cancer Center. And Dr. Oesterreich, Nancy Davidson, and Otto Metzger were the co-chairs of that, and it was supported by the NCI, the Breast Cancer Research Foundation, the Susan Komen Foundation, and other groups. But this was really the catalyst that kind of started things off. A lot of the new research was presented. It was the first ever scientific symposium on lobular breast cancer. And this also coincided with the beginnings of coordination of patient advocates. There was a Facebook group and a breastcancer.org thread. And so word got out about this conference, and the conference organizers really built in and worked with bringing patient advocates in to be full participants in this conference. And 30 patients from around the country flew into this first symposium. And as patients, we were just thrilled. We were so thrilled. There was finally something for us and something that was talking about our disease. And we came out of that very energized and motivated after lots of good meetings. One of the things is that we realized that there were a lot of mutual needs. One is that patient advocates and as patients, we really were starving. We really needed good quality information about lobular breast cancer because there was very little out there and really nothing to help patients and to inform our own self-care. Even things down to the basics, like where can lobular breast cancer spread and what are the symptoms to report to your doctor? So they were really starting from zero on basic information about lobular breast cancer that was patient appropriate. A lot of advocates had a real lack of confidence in the therapies that were currently trialed on ductal cancers, but maybe less was known about how lobular cancers might respond differently for those. And the advocates were really eager to bridge the gaps and support research to help to raise awareness and help others learn about this disease. Researchers and clinicians, by bringing advocates into this symposium, they recognized that they needed input from advocates to help identify the research priorities and the patient needs to get things going. And they also really needed the patient voice and the advocate voice to help make the case, to help raise awareness and the visibility of lobular breast cancer in the community, to bolster funding, and to really make the case for the need for more research. So out of that symposium, we actually had a meeting there, and I actually raised my hand and volunteered to write a letter. And the letter was basically going to be to unnamed somebodies within the breast cancer world. And we were basically going to tell them, it's like, lobular breast cancer is different and we need to do more research. And somehow this letter morphed into an organization that turned out to be the Lobular Breast Cancer Alliance. We had a volunteer patient advocate steering committee, and Steffi led and recruited a remarkable scientific advisory board. And we launched Lobular Breast Cancer Alliance officially at the San Antonio Breast Cancer Symposium in 2016, and things have just really taken off from there. Next slide. So it's helpful, I think, to go back and just do a quick introduction about what is a patient advocate anyway, because I know we have lots of patients and people who might be interested in becoming an advocate on the call, and also maybe some researchers and clinicians who have never really worked with an advocate before. So the definition of a patient advocate is a patient or a former patient, a caregiver or a supporter who wants to learn more about lobular breast cancer and then use that knowledge to benefit others. And that can be through advancing research, education, or support. And there are many, many ways that an advocate can contribute to this mission and goal. Some of the ways can include education and visibility within their community or online, outreach to organizations and institutions, peer support, basically volunteering to help other patients who have lobular breast cancer cope with their diagnosis, policy advocacy, which can be advocacy for more funding, increased funding, against funding cuts or different policies, say for cancer screening, insurance coverage or whatnot. And then, of course, fundraising, which is applicable always across the board, directly for research or in support of organizations. But one of the things I wanted to explain a little bit more about is what And a research advocate is, and this is another type of patient advocate, and basically what a research advocate does is that they can act as a liaison between researchers and patients, and they work directly with researchers to inform questions that need to be answered that would actually meet real patient needs, kind of help with some of the priority setting for research. They can also inform the design of studies and trials, which can maybe help make them be more successful. They can translate and communicate research results back to patient communities. Some of the specific things that research advocates can do is they can partner with researchers to shape research proposals and grant applications. They can review and read research summaries, or lay summaries. They can confirm their interest in a proposal by offering a letter of support. They can participate in consumer grant review, help design clinical trial protocols, and then inform consent agreements. They can work through their local institutional patient advocacy groups or even be part of institutional review boards. They can attend professional conferences and meetings and events as advocate representatives. And then they can also even conduct and present patient-driven research, surveys of patients and that sort of thing. And then share all these results to the broader cancer community. Next slide. So after Lobular Breast Cancer Alliance was formed and we got started, and really for the first 2 or 3 years, we concentrated a lot of our time on just trying to build some basics, put a website up, get basic information up there about lobular breast cancer, because none of these things really existed, and they were certainly not in one place. So we spent a lot of our initial time kind of putting the bones in place for an advocacy movement. And as we did that, and more people found us, Lbca, from around the world, different advocacy initiatives started to pop up. And we're going to meet Claire and Siobhán, who are with Lbc, Lobular Breast Cancer UK and Lobular Ireland, who are doing amazing things over in Europe right now. And then super exciting is the Research Consortium Elbcc in Europe, which has from the start integrated and really embraced advocates and brought advocates into that effort. And so we're going to learn a bit more about that. And all of these advocacy organizations, to different extents, can do the education, raise awareness, and a lot of the activities that are over in that blue box. And everybody, all of these organizations are also still fairly new. You know, we don't have 20-year track records, and people were still figuring things out and building and growing. But it's exciting where all this is going. So one of the things I did also want to point out is another new kind of advocacy that is happening is here in the United States, where there's some local and regional based organizations that are more affiliated with institutions. And so, for example, and the Dynamy Foundation is an example of one of those that is actually doing event-based fundraising and then funding research through the University of Michigan, and now a little bit more collaboratively. And they are, I think they've got something like 6 grants funded right now for lobular research. So we're starting to see more and more of this. I know that there's similar efforts going on at the Cleveland Clinic and at University of California in San Francisco. And I would expect to see more of this type of organizing pop up around the country moving forward. Next slide. So all of this advocacy and energy and the new interest in research as they started to discover the differences and the increased visibility has actually led to some progress. And one of the measures of progress that we feel is important in the patient community is that there are now currently 8 active clinical trials. When I was diagnosed in 2011, there were exactly 0 clinical trials. So there's been some progress here. There's more trials that are coming. And so we feel like momentum is really, at least as far as clinical trials and the things that are going to help refine therapies, it's all moving in the right direction and very exciting. We all notice there seem to be more posters. There's more people talking about lobular breast cancer. There's more presence at conferences and meetings. We still have a long way to go, but it's exciting to see the increased visibility and inclusion of lobular breast cancer and to feel like we're moving in the right direction. Next slide. So one of the things that we did in this paper is we tried to identify some of the challenges in advancing lobular breast cancer research, and then how researchers, clinicians, and advocates could work together to try to move beyond these and make real progress. And there's a much more detailed discussion about a lot of this and specific thoughts and ideas of what can happen, and that's all in the paper. But we broke it out into 8, we called them challenges, and then talked about different opportunities to move forward with it. A couple of these that I will just point out as specific things that are most important to patients is the challenge to define lobular research priorities. As patients, we care a lot that these research priorities go beyond just being an interesting idea. We also really want them translatable into the clinic and to do the things that will help identify new targeted therapies or refine existing therapies that are currently applied to patients. And one of the proposals moving forward that I think Steffi can talk about later is we're looking at a survey, a global survey that could help with researchers, clinicians, and advocates that can help identify and maybe help come to consensus on what some of these research priorities should be moving forward. Another priority that is particularly meaningful for patients is designing clinical trials focused on lobular breast cancer and sharing the trial participation and opportunities for patients to participate and to assist with accrual for those trials. I often say that being a patient with lobular breast cancer is sort of like being the square peg being pounded into the round hole. And sometimes it fits, and sometimes you can make it fit, and sometimes it just doesn't work that well. And what we really would like to do is try to get some treatment guidelines and have a good understanding of what is the best treatment for patients with lobular, and to power some of the big studies and the big clinical trials appropriately, so that we can learn something about lobular breast cancer on these areas where we have the big questions, and also to support additional clinical trials to advance some of these new therapies that would be applicable for us. And another challenge and opportunity I want to highlight is the education, communicating and educational and research information. This is an area I think that advocates really bring in a tremendous strength. Advocacy organizations, through their websites, through their email lists, through social media, through the ability to work with the media, we can actually talk to a very large audience of people, and we can communicate research results, we can communicate needs, we can foster conversations about, within research environments, as also and more broadly to the lay public about lobular breast cancer. And this is, through social media, we can reach not only targeted and stakeholder audiences like patients and researchers and people within the cancer communities and other advocates, we can also actually, take the conversation out to the general public. And so I think through many of these priorities and opportunities, that is one of the areas where advocacy organizations can really contribute the most to moving things forward. And finally, for the all-important research funding questions, what we're seeing now is advocacy organizations are beginning and already starting to actually raise money and actually create new funding sources specifically for lobular breast cancer. And this is something that I think will just continue to grow. But then the other thing that's important and is to actually work within existing funding sources to try to leverage higher proportions of funding for research that goes to lobular breast cancer, even proportional funding according to our histology, so that there is 15% of breast cancer funding through an organization. What about having that dedicated to lobular breast cancer? And also just working to try to, with the existing funding organizations, to really try to designate more funding resources towards understanding lobular breast cancer. Next slide. So in conclusion, so recent progress in understanding lobular breast cancer's differences has really catalyzed research interest, and that has met with new patient advocate organizations that are driving interest, urgency, and funding, and are available and able to do the communications and education piece on lobular breast cancer that was missing before. And what we are hoping moving forward is that there will be a collaborative, multi-institutional, and international efforts that include advocates that can overcome research challenges and help advance therapies for patients with lobular breast cancer. Next slide. So how can we move forward? If you're a patient or an advocate, or actually anyone else, we really encourage you to connect with an advocacy organization and find out how you can get involved and what you can do to be part of all of this effort. And lobularbreastcancer.org, which is the website of the Lobular Breast Cancer Alliance, they have a whole section on that with resources that have what research advocacy is, how to get involved. They have videos and all kinds of things. So that is an excellent place to start for anybody who wants to learn more about advocacy and how to engage in patient advocacy. For researchers and clinicians, what we really just encourage you, as patients, to initiate and support lobular breast cancer powered trials and lobular specific trials and studies. It's just so important. And we hope that you will include and be a partner with advocates. We're not that scary. And we would love to work with you on this. And we also really, really appreciate it so much when researchers and clinicians actually be a voice and speak up within their colleagues and all these meetings that advocates can't get into, but you get into, to really be a voice and speak up to including lobular breast cancer. I was at a meeting a few weeks ago, and a researcher I'd never met spoke up and made the case and really advocated hard, and it was great. It was great to hear someone making the case for more lobular research and why it was important. And it wasn't us, it was others. And I think that's how we're going to make a difference and advance this in the future. So for next steps, some things that we've been talking about that we can probably address more in the conversation is there's discussion that Steffi is familiar with to field a global survey to identify research priorities. There's also efforts to standardize ILC diagnosis, and this is something that Elbcc and the Lobster Pot Initiative are working on, and Patrick and Christine can talk a little bit more about that. And then a lot of interest in how to move forward is discussing a collaborative international ILC research strategy and a framework that would include researchers, clinicians, and advocates, and what would be the next steps moving forward. And I think the next gathering for the ILC Symposium is scheduled for Europe in 2022. And so I just want to say thank you so much to all of the co-authors on this paper and the commentary that we did, and thanks so much to the editors of the special edition where this was published. We're just so pleased to be able to do that and to be able to put this out there for the world. And then also thank you to those who helped with the paper, and to all the organizations who were part of creating this commentary and paper. And now I just want to turn it over to kind of a meet the ILC Organizations Panel, and then we'll move into a Q&A. So briefly, joining us on the panel, we have Christine Desmet, who with KU Leuven, who was with the European Lobular Breast Cancer Consortium, and Patrick Derksen with UMC Utrecht, also with the European Lobular Breast Cancer Consortium. Siobhán Freeney is a patient advocate with the European Lobular Breast Cancer Consortium, and she's also the founding member of Lobular Ireland. Claire Turner is the founder and chair of Lobular Breast Cancer UK, and she is also a Elbcc patient advocate. And then Laurie Hutchinson is the executive director now of the Lobular Breast Cancer Alliance, and she stepped in to replace me, who has retired from that role and is just doing an amazing job. And we have also Otto Metzger, who is the new chair of the Lbca Scientific Advisory Board. So, so thanks so much, everybody, for being here.
Okay, great. Thank you, Leigh. I will stop sharing, and I think I will give it over now to Patrick. Is that correct, Dr. Derksen?
Yes, thank you very much. And thank you very much for all the information about the fantastic work that Lbca has been doing in the States. So I've been asked to give a short introduction on what we've been doing with the Elbcc. And if you will allow me, I have some slides I can share. Just a couple on what our plans are with the Elbcc and how we want to collaborate with the rest of the world. So you can see my slides correctly? Yep. Okay, so it's a bit different in Europe because the Elbcc started based on the research and the clinical work that was done. And then later on, we involved the patient advocates. So really, I started this consortium out of pure frustration because it was so difficult to get all the basic information we got out of the laboratory into the clinical setting. And so the general idea was to get everyone together who was involved in ILC and improve that. So here you see our newly designed logo of the Elbcc, and next to it is a smaller logo from the Lobster Pot, which is a spin-off initiative from Elbcc and is now funded by COST, which is a European organization. They don't provide money for research, but what they do is give money to organize meetings. We can design a professional website, which is going to be live either next week or the week thereafter, and design things like a logo and basically get everything up and running. So general idea, which may be a bit romantic and naive, but what we wanted to do is bridge all these scientific disciplines together. So bridging the actual basic science encompassing biochemistry, genetics, and cell biology, the more translational work that had been going on in mice and in pathology and drug testing, and really couple that to patients in the clinic so they can benefit from what is going on in the laboratories. And of course, we want to encompass all the patients and the patient advocates at an early stage so they can be involved in decision-making on how we should do our research and how we should implement these findings in a clinical setting. So we started in November in 2018. And the first thing we did, we had a couple of brainstorm sessions on how to define the problems within ILC. So these are a couple, excuse me for the slide with all the text on it, just focus on the bold letters. So the first focal area we defined was we needed multi-center data collection, storage, and analysis. So that is a big problem in Europe, and I reckon in the States as well. Maybe in Europe it's even a bit bigger. Because we have all these different countries that speak different languages and usually are not too collaborative. So we want to change that. So that's one thing. The second and the third thing are defining heterogeneity tools for improved diagnosis. So Matthias Christian was talking about this, and it's a hot topic within the field. What is an ILC? How you define an ILC? And how should patient be included for ILC-specific trials? So we need to define that on a global scale. And for that, we need to identify prognostic and predictive biomarkers. So again, it's a collaboration between the basic scientist, the translational scientist, and the clinic in order to come to biomarkers that are actually predictive and prognostic. The fourth thing that was identified as a major point is we need to increase the quality and the quantity of the ILC models. So for too many years, we have been fiddling around with 1 or 2 cell lines or 1 or 2 mouse models, which may or may not be predictive. The opinions are out there, but in any case, we need to increase the quality and the quantity of this, deriving patient material, sharing material, even before it's published within the community so that we get onto targeted intervention trials. And finally, coming to the clinic, we have to define and characterize dormancy. This was identified as a big problem because of the long-term latency relapses that occur in luminal A and specifically in lobular. And we need to develop intervention strategies based on the characteristics that define ILC. So the major themes are centralized data collection, repository, and storage. So what we want to do within Europe, and of course involve the Americans and the Canadians and everyone else, but we start in Europe by creating a website, a CBio portal type setup, where the collection within the different countries is done, but then centralized, at least in silico, so that they will be available for the Elbcc members. We need to set up retrospective and prospective trials, increase the sampling, and get standard operating procedures for analysis, biomarkers, and lab resources. And this is essential that across the ILC field, we standardize the way we look at the samples, we derive the samples, and how we interpret the data that is coming from the samples. Now, the prospective sampling is really important because we not just want paraffin-embedded tissues or tissues that are frozen in the -80. We need viable cells. We need live cells in order to do functional assays. And in that way, we think we can better define why these cells are chemo or endocrine resistant, what defines their long latency relapses after dormancy. And maybe that can also then help to optimize the imaging, which is still in need of optimization and improvement. And of course, a very important point is, as I mentioned, the outreach and patient advocacy involvement. Again, we have Europe, so we have a lot of different countries. So the UK and Ireland are really up and running already. We have a patient advocate in the Netherlands who is very involved for the Untitled. There was a Belgian advocate who sadly passed away, but we're trying to increase this to the Eastern European countries and the rest of Europe as well. And of course, we need a lot of money for this, as everyone else does. So that's a brief overview of the Elbcc. We are on Twitter, so #away if you want to involve us in your research findings and your other events you might be doing. Talking about events, as Leigh was mentioning, we are planning a meeting for the Elbcc and Lbca, and I think we have decided to try and organize this in November of this year. depending on COVID restrictions and other meetings. So keep your eye out on Twitter and our website, which is elbcc.org, for information on the upcoming events in November. So thank you very much. That's what I wanted to say about the Elbcc, and thank you for giving me this stage.
Great. Thank you very much, Dr. Derksen. I think the next, maybe Siobhán, if you can follow up a little bit on discussing the role of advocacy within Elbcc.
Yes, certainly. Thanks, Dr. Oesterreich. So I began, I suppose, my interest, I should say that I was diagnosed in 2016. And you heard from Leigh earlier. When I was diagnosed, I could find virtually nothing relative to my diagnosis. So when I first found Leigh and Diane and Dr. Steffi and the Pittsburgh ILC Symposium, I was just kind of elated and fascinated. And so, you know, you were the catalyst for me to start an awareness and education. I suppose on social media was where I began, really, because I wanted to share this. I wanted other women coming along who were diagnosed to know that they weren't alone, and that there were people out there advocating for personalized treatments and outcomes for women with lobular. And there were lots of researchers out there who had written papers that, you know, were now becoming accessible, and that was hugely important to me. So I started Lobular Ireland in about 2018. I had already been advocating for breast density awareness, and I suppose it was just a natural thing then that I would continue what I see as the original lobular awareness comes from Pittsburgh, and we will always owe a huge debt of gratitude to you, Leigh, and to other people. So that's where I came from. I was invited very kindly by Patrick, Dr. Derksen, to come to the 3rd European Lobular Breast Cancer Consortium, which was held in Leuven. And I was absolutely thrilled to go and represent not just women from Ireland, but I spoke very briefly about patient advocacy and met several advocates. And we were welcomed with open arms. The other thing I would say is that all of the researchers throughout, and there were 2 days, it was fascinating, but all of the researchers greeted us with absolute delight and welcomed our involvement right from the get-go. So that's been going on now for quite some time. And Patrick didn't mention it, but I'm going to just hold it up very briefly. We've been working on a patient information leaflet. Some of you may remember Dr. Karen von Balen from the patient advocate sessions that we had on the very first day. And Karen has just spent so much time with us as patient advocates, and we have something that we think is going to change the landscape for newly diagnosed patients and also clinicians, that we want to get that out there right across Europe. So our goals as patient advocates within the Elbcc are Leigh hit on most of them: to educate, to raise awareness, to build partnerships, to collaborate, to foster relationships with researchers. So I would say that already I'm currently working on ILC grant applications with researchers from the Netherlands and Switzerland, and also one which hasn't started, and it may not be purely the ILC, but it certainly will hit on the edges of it, and that would be something in Ireland as well. Personalized treatments, I think, and screening for women with ILC. So those are our goals that we hope to reach on. And Susan G. Komen also, again, has been mentioned, but through their Advocates in Science program, they've brought a lot of us together, and I've learned a great deal from them. The other thing I would like to say, I would like to pay tribute to somebody who hasn't been mentioned just yet, I don't think, but Diane Mapes. Diane was one of the original team in Pittsburgh, and I know she's a very good friend of most of you here. But Diane began a private Facebook group, which I again found shortly after the symposium, and it was just, for me, it was a lifeline. And I think for many, many women, it has become a very safe space where you can reach out to other women with very similar diagnosis. It's evidence-based, it's scientific, it's not a space for sharing anything other than information, which is moderated very, very, very well. And I've become one of those moderators. So I'm learning a lot about how to moderate from people who've been doing it for many years. But just to say, there are 4.4 thousand women with lobular breast cancer on that group. And then there is another group for lobular women who have become metastatic. So, there is a lot of information out there. The Lobular Breast Cancer Alliance as well, we've started collaborating, and we have meetings with them. And really, I just want to pay tribute to all of you. And I think I've said enough on that. Lobular Ireland is there. Our information is up there. I'm very happy to talk to anybody across Europe, Ireland, and look forward to the next meeting in Europe, hopefully face to face.
Yeah, no, Siobhán, that's great. Thank you so much. And maybe next we can hear from Claire, the founder of Lobular Breast Cancer UK. Claire, you're still on mute.
I'm muted. Thanks, Steffi. I think the story of Lobular Breast Cancer UK is one absolutely of collaboration and partnership, everything we're talking about today. So I was diagnosed in 2017, couldn't find any information anywhere in the UK, found Lbca and reached out to Leigh and Laurie, one of the founders of Lbca, who've been incredibly supportive. Having a background in charity work, I know that partnership, we're so much stronger when we do things together. And so I reached out and now work with 9 amazing women who are all people diagnosed with lobular breast cancer who are working together to found Lbc UK. We're a registered charity. We cover the 4 countries of the UK and their different health services. So a bit Patrick was mentioning about Europe. It's not as complicated as Europe, but what we have a different language. We have Welsh. We have completely different health services with treatment protocols as well. So we've kind of got that happening with ourselves. Our key long-term purpose is that everyone diagnosed with lobular breast cancer lives and lives well. We're slightly late in launching. We would hope to launch last year. I don't know if anybody has ever tried to set up a bank account in a pandemic. It's taken us 5 months, so we're slightly delayed, but hoping to launch next week. But the key thing, I think, with Lbc UK, we're patient-led, and all our plans and all our activities are going to be guided by the needs and the issues that are raised by the patient. So what the key 4 areas we're looking at focusing on, particularly for the next 12 months, is information. The one thing we hear from all patients is they don't have the information. Often they're not even informed. They're told they have breast cancer, not lobular breast cancer. One of the things I always make sure I do is say, no, I have lobular breast cancer. I don't just use the generic term. So all information, again, is going to be scientific, evidence-based as well, which is really important. And we're working with a scientific advisory group, one of whom, Val Brunton, actually is presenting some of her research here at the conference, to ensure that all the information we put out is accurate, it's based on the latest scientific research. But with the SAG as well, it's also a really good way for us to get information out and involve researchers and clinicians who might not be part of the Lbcc yet. And quite a few of our scientific advisory group aren't or don't know about Lbca. So we as patients can share that information back. Patient advocacy is going to be absolutely key. We want everyone to be their own best advocate, to go into those meetings with their medical teams with the information they have. We'll be looking at research advocacy, so we can ensure we've got trained patients who can work with researchers and clinical researchers on developing new research and participating in trials, and also looking at how we can change policy and treatment protocols as well, which, as I said, are not standardized, and somebody gets MRI for monitoring and somebody in another location doesn't. How can we really standardize those treatment protocols based on the research that all of you are doing? Research will be key for us as well. And one of the things we're going to be launching with is just doing a bit of research across the UK to find out, as I said, we're patient-led. So we want to find out what are the key issues and concerns of patients here in the UK. And particularly one of the things we want to look at, we're working with De Montfort, and one of our trustees is actually a health psychologist at the university. What are the psychological impacts on lobular, which seem to be additional than standard breast cancer, due to a potential for late recurrence, but we're only monitored for a short period of time. And also, the imaging has been raised as another concern. And we want to be working with researchers across the UK and internationally to develop new research and trials. And part of our job we see is taking the work that you are doing as researchers and sharing that with patients across the UK, and sharing it in a way that is clear and they can understand, and supporting you in your public engagement work for your research. Support is another area, but one of the things we want to emphasise is we're not a professional support organisation. There's lots of organisations that do that in the UK and internationally. So we've been talking with them to make sure, for example, breast cancer now, we've had a conversation. Do all your nurses on your helplines know about lobular? How many patients have you got that are working in befriending systems that have lobular? We've been speaking to the Royal College of Nurses. Do all your nurses know about lobular? So those are just kind of the 4 key areas that we're looking at focusing on. And as I said, collaboration is one of our core values. So it was fantastic to be invited to be part of this paper and this panel discussion. And we'll keep working with, obviously, Elbcc, Lbca, research institutes, other patient advocacy organisations and groups. So anyone who's here from the UK, please get in touch. I've gone through all the people at the conference and sent you all a connect if I've noticed you're from the UK. We'll be on Twitter and Facebook. We'll be launching, hopefully next month, with a press campaign. So we're just really excited to be part of such a dynamic group of researchers and clinicians and organisations, and so we can make real change for those patients coming after us as well. So thank you for allowing us at LBC UK to be part of this.
Great, thank you so much, Claire. And last but not least, Laurie Hutchinson, Executive Director of Lbca. And Laurie is going to share some slides with us, yeah?
Yes, thank you. Can you see that?
Great.
Wonderful. So thank you, Leigh and Steffi. I want to say how grateful I am that Lbca already existed in 2018 when I received my ILC diagnosis, and how happy I am to be working to ensure that all the founding advocates and researchers' vision is realized. And I also want to thank you, Steffi, for being, as Siobhán referred to, I think in the orientation session, as our lobular mother. And continually fueling more research on ILC. So thank you for that. I joined Lbca as a volunteer, and I was so quickly inspired both to learn about the basic science of breast cancer and lobular breast cancer, and learned, like Claire, to say, I have lobular breast cancer, and why that matters. And it led me to really want to pitch in, and then ultimately when I talked with Leigh about her departure, to help Lbca keep going. So over the past year, just touch on a couple of things. We've taken major steps to become a sustainable organization, which entails fundraising and also continuing to get the word out. We have a growing number of staff. We now have a paid team. We are striving for our own tax-exempt certification status and hope to achieve that by the year end. We have a growing number of active volunteers, and we have expanded our International Scientific Advisory Board of ILC experts, which includes Steffi and Dr. Derksen and Dr. Metzger. So it's a wonderful group, and it's international. We have a comprehensive website, and we have recently revised it so it's more navigable. And we've added some sections and added some depth, and we want it to be something not just for patient advocates, but for scientists and researchers as well. We have a current publication library of ILC research. We have a web page dedicated to information about clinical trials, and we keep all of this information updated. We have a For Patients page with FAQs and information sheets about lobular breast cancer with graphics. And now we have designated a section all about metastatic lobular breast disease. We also have tools, I think Leigh mentioned. We have tools for research and education advocacy. We have recorded panel presentations and webinars from conferences. Webinars done specifically to train patient advocates, but also just about controversial topics or where the research is in lobular breast cancer research. And we really do try to help anyone interested connect to other training so they can become research advocates if they'd like to be at the table, focusing on making sure that more breast cancer research is targeted to the 15% of lobular breast cancer disease. We strive to get the word out every day about ILC via social media. I think our communications director is tweeting madly from the conference today, and I'm very appreciative. And we've really grown our capacity to be able to do that, and it's really important. And I know Lbca, I think, is credited, and I think it's appropriate for really raising awareness across the advocate and research community about ILC. And we've just started to fund ILC research directly. We were able to raise enough money this year to issue 2 grant solicitations with the administrative help of the AACR and ASCO. And this is really a first step for us in our plan to have an ILC research division. And with that, we will be able to promote, solicit more early investigator and multiple year ILC research grants. We are going to promote cross-lab and cross-country collaborations, which really is required for ILC research. And we are aiding in the development of ILC research models. And just this week, we were talking with both the ELBC members, with Dr. Derksen, about how to collaborate and figure out the best strategy for developing more research models. And then later in the week, we spoke with National Cancer Institute, which they've invited us to provide expertise, and they will provide and develop some organoids. And so we're very excited about all of these plans underway. And again, through collaboration is the only way to make it happen. So just briefly on the key challenges that Leigh and Steffi mentioned in the paper, I just want to touch on a few things. We really rely on the expertise of our scientific advisory board members, and we solicit patient advocate perspectives to determine our ILC research priorities. And we will be contributing to and have contributed to designing the survey, and look forward to the results that Steffi and Leigh talked about earlier of the survey of scientists, researchers, and advocates worldwide. We share current information on clinical trials focusing on or enrich for patients with ILC and encourage participation. And we believe it's key to raise awareness and educate patients and researchers all over the world about the most current information about ILC research. And I'm just seeing the time, so my last comment is we seek to promote opportunities for collaboration with ILC-focused advocates, scientists, researchers all the time, all over the world. And I'm really happy to have met all of my colleagues on this and to be able to work with all of you on doing that. Thank you.
Great. Thank you so much, Laurie. I think these were really, really great presentations, great introduction to the different organization. I do know that, we are running late, so we don't have time for Q&A, but I think that's fine. I think it was really important for all the organization to introduce themselves. And, Patrick, that might be something, at the next Elbcc meeting, maybe the topic can be picked up, and the discussion we wanted to have now, we might want to have then. So that's one idea, right? I mean, maybe continue it at the next meeting. Yeah.
Excellent idea.
Yeah. So thanks everyone for presenting. Really appreciate that. And I think with that, I give it over to Patrick to run the first scientific session this morning. And I think all of us should log out from this and log into the main site. Okay, thanks again, everybody.
So I'm under the impression that I'm at the main stage. Are you as well, Matthew?
I hope so.
Yeah, okay. Because Steffi said we need to log off and then log on again, but... Nadine, are you around?
You're good, Dr. Derksen. You guys can go ahead.
Okay, excellent. Okay, so welcome everyone to this session, which is the first of the two biology-focused sessions of today. My name is Patrick Derksen. I'm an experimental and preclinical researcher at the University Medical Center in Utrecht in the Netherlands, and I will be hosting this session. So this session was first named New Kids on the Block, but given that we have a very established ILC researcher like Matt Sikora as speaker, we thought it would be more appropriate to call it New Avenues on ILC Research. And just a reminder that if you have questions, please put them in the Q&A. So as mentioned, our first speaker today, Matt Sikora, received his undergraduate degree and PhD in pharmacology from the University of Michigan prior to joining Dr. Steffi Oesterreich at Pittsburgh for his postdoctoral work. So he started his lab at the University of Colorado Anschutz Medical Campus in 2016, where he focuses on the biology of ILC and ovarian cancer. So Matt has done seminal work on ILC, studying the role of estrogen signaling and endocrine resistance. And his talk today is titled Understanding ILC as a Unique Context for Interaction Between Estrogen Signaling and DNA Damage Response. Matt, the floor is yours.
Great. Hopefully my slides are up okay.
Yeah, looks good to me.
Great, thank you. Well, thank you very much, Patrick, for the very kind introduction and for the invitation to speak today. I'm really excited to share our recent work on estrogen signaling in lobular carcinoma. So we've had a lot of great talks yesterday, and even this morning our session touched on the idea that understanding estrogen signaling is not a one-size-fits-all approach, especially when it comes to ILC. And as a field, this shouldn't surprise us because we do know that estrogen and the estrogen receptor, or ER, do different things in different tissues. Obviously, the physiological needs of the bone versus the breast, for example, are very different when it comes to estrogen. But we're taking the approach that ILC represents one of these different tissues, in fact, and that's in part because it has unique risks associated with estrogen, for example, from hormone replacement therapy, and very distinct anti-estrogen responses that we've talked about already, and I'll touch on briefly in a moment. I think it's important to remember also that most of our understanding of how ER works and how it functions is based on ductal cancer biology, and in fact, mostly the cell line MCF7. What we're learning more and more, and that we've already talked about, is that ER and ILC is far more than just the positive nuclei in our diagnostic IHC. And my lab is taking the approach that by understanding how ER works in ILC, we can build the foundation for better anti-estrogen-based treatment strategies for patients with ILC. So we've already talked about that nearly all ILC express the estrogen receptor, and most fall into the luminal A molecular subtype. But as we're discussing, this really isn't the whole story. In part of these curves, we've talked about several times already that ILC tumors just aren't responding the same way to tamoxifen as other tumors, which to us is a suggestion that estrogen receptor function is just different in these cells. They're responding to anti-estrogens differently than other breast cancers. Around the same time, we were learning from model organisms, the model cell lines, in this case, the MDA-MB134 cell line, that they are in fact treating the SERM class of drugs like partial agonists or weak estrogens. And this partial agonist activity, in fact, we're learning extends to some of the next generation oral SERDs or downregulators, such as AZD9496. So it's really a very broad class effect that estrogen receptor in ILC is seeing these anti-estrogens as weak estrogens or partial agonists. And it's not just the MM134 cell line. We're seeing this in other models, including the PDX HCI013. And other forms of SERM resistance have been reported in other ILC models, notably the SUM44PE work from Becker-Riggins' lab. But altogether, my lab is taking the approach that these distinct SERM responses suggest that estrogen receptor has distinct functions or regulators in ILC cells. And we think that the prime candidate for ILC-specific estrogen receptor co-regulators are actually transcriptional co-regulators. And these are the proteins that work with transcription factors like estrogen receptor to either open or close a local chromatin and turn a gene on and off. And importantly, we also know that cell type specific co-regulator function or expression can mediate cell type specific SERM responses. The current hypothesis in the field is that in general, if there are more activating type co-regulators expressed, this would allow estrogen receptor, and in this case, tamoxifen, to turn a gene on, perhaps like in the bone where we know tamoxifen acts as an agonist. But instead, if you have a more repressive like cohort of co-regulators, this would allow the gene to be turned off, which may be more like the breast where we know TAM is an antagonist. So we're asking the question, is ILC in fact a distinct cell type that expresses its own distinct cohort of estrogen receptor co-regulators, and that this defines how ILC cells are responding to SERMs? To understand the ER partner proteins in ILC cells, we use the immunoprecipitation mass spec technique RIME, which is rapid immunoprecipitation and mass spectrometry of endogenous proteins. This was developed by Hisham Mohammed and Jay Carroll's lab. And essentially, it's a modification of CHIP that it relies on fixing protein complexes prior to the immunoprecipitation. It keeps all of the complex together during the IP process and lets us get a better handle on who's actually working with estrogen receptor. So we performed estrogen receptor RIME from 3 ER-positive ILC cell lines, the MM134 model, 44PE, and BCK4, and we identified a whole host of ER-associated proteins. We compared this then to the original data from the IDC models, MCF7 and ZR751 from the Carroll Labs paper, and we identified 73 shared proteins, and this included, happily, the estrogen receptor itself, along with proteins like FOXA1, GATA3, NRIP, etc. And this gave us a lot of confidence in the experiment itself, but also suggests that there is a core co-regulator machinery that is shared across these breast cancer contexts. But certainly we're very interested in these ILC-specific hits, and this included families of chromatin remodelers, mRNA splicing factors, DNA synthesis and repair proteins, and so on and so forth. And we've got more data on all of these hits in our recent study. But we needed to understand which of these 100 or so factors were actually most critical for estrogen receptor function. And to do this, we performed an siRNA screen using the MM134 cell line. And we targeted about 200 factors from RIME, as well as some other in silico predicted co-regulators. And we looked for the ability of these target knockdowns to suppress either estrogen-induced growth or tamoxifen-driven growth, that weak agonist or weak estrogen effect. And somewhat gratifyingly, the top hit in this screen was estrogen receptor itself, which is a great positive control that we can suppress the system. Also among the top hits was the protein FOXA1 that Dr. Jesselson discussed yesterday. So this is another great positive control for this screen. Aside from estrogen receptor itself, though, our top hit was this gene MDC1. And I'll tell you more about what MDC1 is in a few moments. But what we found in this screen is that knockdown of MDC1 suppressed estrogen-driven growth in the pink here. It suppressed tamoxifen-driven growth. But it didn't affect androgen-driven growth, in this case by a synthetic androgen. We've learned that this particular cell line can use AR to drive growth independent of ER, but this tells us that MDC1's effect on growth is specific to estrogen receptor. And we found in further study that MDC1 knockdown suppresses growth in ILC cells, but not in similar ER-positive IDC cell lines, or ductal carcinoma. We confirmed the ER-MDC1 interaction by classic co-immunoprecipitation methods, but then we use this method proximity ligation assay, or PLA, to screen additional cell lines. And how this assay works is it uses 2 antibodies for our targets, ER and MDC1, and if they are in physical proximity, you get this little light up, which is a yellow dot in the blue nuclei. And what I hope you can appreciate is that in the ILC cells on top, we see the little Christmas tree light ups suggesting that, or showing that ER and MDC1 are in close proximity and likely interacting, but we don't see this in any of the ER positive IDC cells on the bottom here. Also, we found in an ER RIME data set from Mitch Dowsett and Leslie Ann Martin, they used ER RIME in the 44PE model as well as the 44PE ER mutant LTED model. And in their data, MDC1 was also among their top hits for estrogen receptor association. So altogether, this leads to our hypothesis that MDC1 is critical for estrogen receptor function in ILC cells. So since MDC1 was critical for estrogen-driven growth, we then looked to see what role it plays with the ER transcriptome. And we did this using two ILC cell lines, the MM134 and MM330, and compared that to the ductal model HCC 1428. And I'll walk you through this experiment. What we did was we hormone-deprived cells and then knocked down either a control construct, we knocked down estrogen itself, FOXA1, or MDC1. We then gave the cells back estrogen and looked at estrogen-regulated genes and how these were impacted by target knockdown. So what you can see on the leftmost column here in the MM134s is our no estrogen baseline, so that's when the system is off. When we add estrogen back in the control setting, it turns on a whole bunch of genes in yellow and turns off a whole bunch of genes in blue, roughly 3,600 genes in this MM134 model. If we knock down estrogen receptor, all of these get blocked, confirming that they are in fact ER target genes. What we found with FOXA1 and MDC1 knockdown, though, is that we had a series of genes that were regulated by neither of those two proteins, some that were controlled by FOXA1 only, that is ablated by FOXA1 knockdown, some that were controlled by MDC1 only, or some that were sensitive to knockdown of either of these factors. And altogether in MM134s, more than half of the ER transcriptome was MDC1 dependent. We did see similar patterns in the other ILC model as well as IDC, but to a somewhat smaller extent regarding MDC1 target genes. When we did more pathway analysis of the ER-MDC1 target genes, we actually found that estrogen response hallmarks, including the estrogen response signatures and MSigDB, were enriched in both ILC and IDC cells. But importantly, we saw a whole series of DNA repair signatures that were enriched only in the IDC cells. And this suggests that whatever MDC1 is doing to estrogen receptor function is restricted to impacting DNA repair. And I'll get back to that in a minute in MDC1's role in DNA repair. We then looked further at these MDC1 target genes and looked at the regulatory motifs that are in the promoters of these genes, that is the sequences that are probably responsible for directly regulating these genes. And among the ILC-specific MDC1 targets, these were enriched for estrogen receptor motifs and related half EREs and other nuclear receptor motifs. Whereas the IDC-only cell lines were enriched for E2F motifs, consistent with that role in DNA repair. So taken together, we think that MDC1 has a unique role in ILC cells in regulating the expression of ER-driven genes and hormone response element-driven genes. So I've said MDC1 a whole bunch, and now I'll finally tell you what it is. MDC1 is mediator of DNA damage checkpoint 1, which normally is a cornerstone of DNA damage response. And what it does is that upon a double-strand break in the DNA, this histone variant H2AX gets phosphorylated to form gamma H2AX. And MDC1 in that setting can then bind gamma H2AX, recruit more ATM kinase in the mRN complex, recruit double-strand break repair proteins, and initiate downstream DNA repair signaling. In that context, as a critical DNA repair protein, MDC1 loss is actually quite common in cancer. A recent review highlighted that across solid tumors, MDC1 expression is typically reduced or lost altogether in tumors compared to normal tissue. And this is particularly true in breast. There aren't a whole lot of studies, but several show that high-stage breast cancer or those with associated nodal metastases have a higher incidence of MDC1 loss. And this is likely because MDC1 loss is going to induce genomic instability and increase tumorigenesis, and in this setting, analogous to what a BRCA1 or BRCA2 mutation would do in causing dysfunctional DNA repair. But based on our data, we think that maybe MDC1 has a different role in ILC cells. To start to get at that, we looked at MDC1 expression by immunohistochemistry and tissue microarrays. And just what I'm showing you here are some representative images of MDC1 staining and ER staining in ILC. In the ILC cells, we saw some tumors that had fairly homogeneous expression of MDC1, pretty consistent cell to cell, and others where we had pretty heterogeneous staining. You can see cells that are 0, 1+, 2+ across this bottom tumor here. IDC tumors looked a little different. We saw a lot more tumors that had very intense, very strong MDC1 staining, and we saw a whole bunch that had absolutely no detectable MDC1 at all. And when we looked across the cohort we had access to and broke this down by histology and ER status, we saw that overall the median or mean MDC1 H-score wasn't changed across different tumor types or normal non-malignant tissue. But what we did notice was that there was the distribution of MDC1 staining in ER-positive ILC looked a lot more like normal non-malignant tissue than it did relative to IDC or even the few ER-negative ILC we had. And using this lower quintile as a cutoff, it turns out that MDC1 is low or lost altogether in about a quarter of breast cancers, but less than 5% of ER-positive ILC. We just had one tumor of this data set that had lost MDC1 expression. And this suggests to us that MDC1 has a unique role in ILC etiology. Typically, as I mentioned, MDC1 would be lost to promote genomic instability in other tumor types, but potentially in ER-positive ILC, these tumor cells want to keep MDC1 because of its critical role in supporting estrogen receptor function. So based on this, we started asking whether or not MDC1-driven DNA damage response is actually functional in ILC cells. As I mentioned, one of the initial functions of MDC1 is to bind that gamma H2X protein at damage sites, and this actually manifests as damage-induced foci by immunofluorescence when we look at cells after inducing damage. And in an IDC model, if we induce damage, in this case with a topicide, we can see those beautiful foci forming. I got red gamma H2X foci and green MDC1 foci, and when they combine, you can see little yellow spots in these cells. So this is performing exactly as we expect, in that they're forming those DNA repair sites in the genome upon damage. ILC cells, we didn't quite see that. We could see an increase in foci of either of these independently, but when we tried to find localization, it wasn't as strong, and we saw a large number of these cells that just have this diffuse nuclear gamma H2X signal that never really coalesce into foci. And there's a little bit of data in literature suggesting that this pan-nuclear gamma H2X signal is consistent with incomplete initiation of DNA damage response. So the cells aren't quite finishing getting through DNA repair potentially. And there's actually evidence in literature and from TCGA that this incomplete DDR initiation may be happening in ILC tumors as well. So in TCGA, they had reported the 3 different ILC subtypes based on expression, and one of those, the proliferative subtype in the green line here, was associated with poorer disease-free and overall survival. When they then looked to see what features could define that proliferative subtype, one of the stronger features was an increased score for a DNA damage response signature in protein array analyses. And we actually went back and just looked at this DNA repair or DNA damage response signature across TCGA, and a high score for this DDR signature was sufficient to predict poor overall survival in ER-positive ILC, but was not the case in ER-positive IDC. So mechanistically, we have a long way to go to understand this, but one key question then is the elevated DNA repair signature or DDR signature linked to MDC1 dysfunction in DDR or potentially to its novel co-regulator activity in ILC cells. And to start to get at this, we're looking with a really cool novel assay at the single cell level called haircut. So we can combine single cell RNA sequencing with this DNA repair assay haircut developed by the Hesselberg lab at CU Anschutz. And this measures the activity of DNA single strand break repair enzymes. And we're hoping to use this as a surrogate for the double strand break repair pathways that MDC1 typically functions in. But what we did was we used the MM134 cell line, and we did single cell RNA-seq from cells that were hormone deprived in gray here versus plus estrogen in pink. And then we looked at the expression and activity of MDC1 target genes and FOXA1 target genes from our original study. And what I hope you can appreciate is that MDC1 target genes are active in a different subset of cells than FOXA1 target genes. We don't yet know how these two cofactors relate to each other, how they're linked, but this suggests that MDC1 is essentially an independent co-regulator of FOXA1 in this setting. But among these cells based on expression, there were 8 different expression clusters. And we noted that one of these clusters in particular, cluster 5, had low FOXA1 activity, but it had high MDC1 activity. When we looked then at DNA repair capacity in these clusters, Overall, we saw that one particular reporter from the haircut assay that for base excision repair was lower in plus estrogen cells than minus estrogen cells. The activity was decreased plus estrogen. Across those 8 expression clusters, though, here they are ranked from highest to lowest overall ER activity. And despite this cluster 5 having high overall ER activity, it should have low base excision repair activity, but was actually separate. It had among the highest BER activity among all of these clusters versus only this C2 over here. So again, we have a long way to go to understand this mechanistically, but we think that differential MDC1 co-regular activity may thus be linked to or otherwise drive dependence on specific DNA repair pathways, potentially because it's not playing its normal role in DNA repair. So to summarize, hopefully I've convinced you that classifying ILC as ER positive and luminal A is simply missing the whole picture. In my lab, we're taking the approach that this is because estrogen receptor works with unique transcriptional partners, the quote-unquote co-regulators in ILC cells. We identified MDC1, canonically a DNA damage response protein, as a critical ER co-regulator in ILC cells that is necessary for both estrogen and anti-estrogen response. And our current hypothesis is that MDC1 may be advocating its canonical or normal role in DNA repair to work with estrogen receptor. And ultimately, this may cause DNA repair dysfunction. Our long-term goals are to actually define how MDC1 is working as a co-regulator, what that activity is, and what the role for MDC1 is in ILC etiology and tumorigenesis. We'd like to target MDC1 co-regulator function directly to combat anti-estrogen resistance as we learn more about it. And ultimately, we'd also like to exploit this potential MDC1-related DNA repair dysfunction. Finally, thank you for your attention, and I'd like to thank my team. This project currently is driven by Joe Sotnick, a research associate in my lab, and previously by Evelyn Bordeaux, a graduate student in the lab. My collaborators, including at Utah and Georgetown, are helping me move this into new context. Thanks to constant support from Steffi at Pittsburgh. Huge thanks to my Lbca supporters through my ACS funding, Leigh and Laurie. And I'm happy to take questions.
Okay, thank you very much, Matt, for a beautiful talk. Very intriguing data. Just as a reminder, please see any. I can start, Matt, on your differences between ductal and lobular if you knock down MDC1, right? So, wondering, have you any idea on what could be the mechanism? I mean, we looked at ER dependency in ductal versus lobular, the models you're using, and is there a difference in proliferation and so forth? Do you have any insight on that?
So you're asking the difference between MDC1 function in the two contexts? Your audio cut out.
Yes, that's one. So I can imagine less dependent on ER than the lobular one, or vice versa. So there might be differences between the different cell lines if you looked at that.
I'm sorry, your audio keeps cutting out. I'm missing your question. So you're asking if, go ahead.
Yeah, shall I try to readdress the question? So are you hearing me correctly now?
Now I am, yes.
Comparing lobular, and you're using two cells, wondering whether you looked at responsive to the ER the lobular versus the ductal cell line, and whether that also could impinge on differences you see if you do an MDC1 knockdown and compare the responses in these cells.
Oh yes. Oh, so the relative MDC1 dependence. Yeah. So we're still trying to understand relative dependence. So the IDC cells we used, if we knocked down MDC1, it doesn't impinge ER-driven growth at all. The classic MCF7s, it does decrease their growth a little bit, but they also become hypersensitive to damage. So it's clearly linked to DNA repair in that context. And in that context also, and in the literature more broadly, MDC1 knockdown normally breaks cell cycle checkpoints. It's one of its canonical roles is that upon damage, it induces arrest, and that's happening in the ductal cells. In lobular cells, when we knock down MDC1, it actually is causing a G2M arrest. So it's completely opposite what it's canonically doing. But it's more consistent with its role as an ER co-regulator. It's critical for a big part of the ER transcriptome. What we don't know yet is why MDC1 has this novel role in ILC cells. We don't know what is facilitating or driving the ER-MDC1 interaction. We have some hints that there are some novel isoforms of MDC1 in ILC cells. It doesn't explain everything. MDC1 is also heavily post-translationally modified, and there are some putative sites that would either facilitate or prevent ER-MDC1 interaction based on differential post-translational modification. So we have a long way to go. It's a big protein, and it's been a bear to clone and work on, but we're getting there. Hopefully I answered your question.
Okay. Thank you very much, Matt. We have a question from the audience. The question is, could you please repeat or expand your comment about the new SERDs and ILC?
Oh, so yeah, so I mentioned that there's obviously been a huge interest in developing oral SERDs because of the difficulties with administering fulvestrant to patients. So there's probably at least a dozen in literature, and there's been a handful of papers that have shown that a few of the earlier, I guess, first gen, next gen SERDs, like AZD9496, actually had the same partial agonist activity in lobular cells as ductal cells. I think importantly, a lot of the pharmaceutical companies have now realized that they need to include at least lobular models in their screening. And a good example there's a Genentech paper where they had a PDX and 2 ILC cell lines, and they had different responses to the SERDs. So it's helping them guide their development pipelines to choose SERDs that are more broadly downregulators like fulvestrant. So I don't think it's going to be an issue clinically, but the fact that this has happened in the development pipeline, I think just gives us more evidence that ER is behaving differently in ILC.
Thank you very much. Another question from the audience is, do you know if there are any differences in radiation sensitivity if you compare positive IDC and ILC?
I don't know the answer to that. We've been trying preclinically, and the ILC cells seem to be, they grow a little bit slower on culture, so they're not as responsive. And we've had a hard time understanding whether or not the MDC1 loss-driven radiation hypersensitivity that should happen, whether that actually happens in ILC cells because MDC1 knockdown causes cell cycle arrest. So we've had a hard time mechanistically tying those two things together or not. And obviously this should suggest that if MDC1 is not playing its normal role, that ILC cells should be more chemosensitive if they can't repair. But obviously we've discussed that ILC cells are not necessarily chemosensitive or responsive, but this may be because MDC1's normal downstream signaling to apoptosis and cell death after damage isn't happening either. So we're trying to figure out other ways to take advantage of this potential dysfunction and whether radiation is a good approach or other targeted inhibitors in that setting. But yeah, the new role in cell cycle has been complicating how we understand that pathway.
Yeah, fully agree. So I think final and slightly provocative question. Would we theoretically want to block or enhance AR signaling in treating IR?
AR signaling. Yeah, very provocative. I think there's a lot of backstory to that question that is maybe unknown to a lot of the audience. In the limited stuff that we've done in cell lines, we had earlier looked at more hormones beyond estrogen and what they do, and a lot of them have at least some partial agonist activity, or not partial agonist, but they have some capacity to work around ER, independent of ER. In the 134 cell line, we've seen that glucocorticoids or androgens can drive their growth. So I would love to see something like an abiraterone or a broader synthesis inhibitor. Obviously, that's complicated with glucocorticoids, but I'm curious about what antagonists will do in that setting, more so than agonists, I guess.
Okay. Thank you very much, Matt. Great talk. Great responses from the audience. I think we will move on to our second speaker, which is George Somos. He did his PhD at the Foundation for Research and Technology, working on TGF signaling pathways, and then he moved to the EFPL, which is in Lausanne, in 2013 to work in the lab of Catherine Briskin, where he is a senior research scientist focusing on experimental cancer research with a strong. So George has done and is doing wonderful work on developing transplantation-based ER-positive mouse models for ILC, and recently has identified lysyl oxidase like 1 as a very important oncogenic factor in lobular breast cancer metastases. So today, George is going to talk about novel models of lobular breast cancer and what they can do to advance the field. So George, the floor is yours.
Hello, Patrick. Can you see my screen? Can you hear okay?
Yes, I can hear you, and the screen is in presentation mode.
Okay, so I would like to thank Dr. Patrick and the organizing committee. It's my pleasure to participate in this wonderful ILC 21 Symposium and the ILC community in general. So to start off, unlike no special type, NST, breast carcinoma, only few laboratory models exist that fully represent the unique biology of invasive lobular carcinoma, ILC. So this can be attributed to lower engraftment rate, which is typical for estrogen receptor positive breast cancers, the lack of clinical samples with the relative lower incidence of ILC, the fewer ILC patients in clinical trials, and difficulty in obtaining enough biopsies and material for research studies. So now, briefly in the next 4 slides, I will review selected published ILC models, including ILC and non-ILC cell lines, ILC organoids, genetically engineered mouse models, patient-derived and ILC-derived cell line xenografts, and other animal models. So to start off, cell lines are really viable experimental tools, but however, ILC is very challenging to establish as an in vitro cell line model. The list of authentic ILC lines you can see here is very short and has limited patient-related clinical and treatment information. And out of more than 100 breast cancer cell lines, only around 7 can be really tracked back to histologically confirmed ILC. So from these, only a handful showing here in the bottom, growing in a discohesive manner, have been really studied more extensively, with few cases derived from primary tumors, but mostly from pleural effusions or ascites. So moreover, although 90% of ILCs are estrogen progesterone receptor positive, only a minority of the available ILC cell lines expresses ER. Finally, the vast majority of the established ILC lines express mutant P53 and have been adapted to grow into the in vitro for extended periods. So now, interestingly, 4 ILC lines have been successfully grown in 3-dimensional matrix and culture, also indicated here on the top right, and with a generally loose morphology. Now, of course, patient-derived ILC organoids, which derive directly from freshly dissected ILC, can better conserve the original genetic status of the primary or metastatic ILC. However, there are less than a handful of patient-derived ILC organoids. In the example shown here, NST carcinomas generally give rise to solid, coherent organoids, whereas the lobular carcinomas generate discohesive organoids that retain estrogen receptor, yet remains to be seen their hormone sensitivity in this setting. So next is the genetically engineered mouse models, the more complex in vivo models that have been really invaluable tools in breast cancer biology. However, with regards to estrogen receptor positive ILCs, their use has been limited in that most of the transgenic models develop ER negative tumors. However, recently, genetically engineered mouse models based on mutations, the most commonly mutated genes for human ILC, CDH1, PIK3CA, or PTEN inactivation, provide new experimental tools to study ILC, along with the first described pleomorphic ILC models, and they can recapitulate different variants of ILC. So lastly, if we want to really study ILC, we should also study the actual tumor ILC cells. And however, with using the conventional approaches, you can see here in the upper right with the injection in the subcutaneous stroma or injection in the fat pad, only few estrogen receptor ILC positive cells can grow, and they mostly require exogenous estrogen, and the estrogen levels that do not correspond to what is seen in women, the most postmenopausal women. So similarly, PDX are very difficult to establish in this setting, and there are very few PDX exist because mostly high-grade triple negative PDX growth, and here is an example of few of them. So now for the next of the slide, I will focus in 3 questions: how we can really improve these preclinical ILC mouse models, how the lactiferous murine and mammary ducts are implicated in these efforts, and what we have learned and what is the outlook. So now we and others recently have shown that using a mouse intraductal mind model, we can study directly the ILC cell lines, also the actual tumor ILC cells that we obtain directly from the operating room, and we can inject them back in the microenvironment where is more physiological, more relevant, and is deprived of stroma cells. It's the milk ducts. So in that way, we can take patient tumor cells and study them in a preclinical setting and see whether these cells are aggressive or not, if they metastasize, where they metastasize, and we can then experimentally test several therapies in this setting. So now, for example, to assess whether this mind approach reflects the biological properties of tumor cells and also normal cells in these graphs, we grafted cells from triple negative, NST, ILC, BRCA1, or either normal cells from reduction mammoplasties. So here in this example, or 3 ILC patient samples established themselves and proliferate, but grew at lower rates comparing to NST or triple negative tumors as is expected, and also plateaued at lower levels. We also see that engrafted breast cells from individuals with mutations in BRCA1 in this graph show the trend to grow faster than normal cells derived from reduction mammoplasties, further supporting the biological relevance of this mind model. So here I just summarize the typical stages of tumor progression in the mind model, which in general, the initial engraftment occurs within the first few days after intraductal injection, mostly by replacement of the mouse epithelial cells, and then it's followed by an extended in situ phase, followed by invasion and metastasis in clinical relevant organ. So using this intraductal approach, we wanted to assess whether the ILC-derived cell lines that I mentioned at the beginning recapitulate the biology, the unique biology of the human ILCs when xenografted to the mouse milk ducts. So here in this example, we tested 2 ER-positive lobular ILC lines, MDAB134 and SUM44 cell lines, which shows rounded morphology here, as you can see on these images, with no detectable ECAD-hearing expression. So to identify specific difference with NST ER-positive models, we also compare directly with MCF7 and T47D cell lines. So all cell lines were infected with a red fluorescent protein conjugated with luciferase expressing glendivirus, have been selected by flow cytometry for RFP expression and injected into the milk ducts of female NSG mice. So in vivo radiance measurements within a week of injection here showed high engraftment rates, more than 83% for the lobular and also for the NST cell lines. Measurement of in vivo radiance here in this graph, you can see that the ILC cells, as expected, had lower growth rates than the non-ILC ER-positive HER2-negative cell lines. The growth curves now of MCF7 and T47D did not differ significantly between them, but differed significantly from the curves of the ILC cell lines. So in particular, the NST cells showed faster growth over the first 4 weeks post-injection. And in contrast, the ILC cells grew at a more constant rate throughout the whole experiment. So now 5 months after injections, the gland that xenografted with NST cells were palpable, whereas the 134 and some 44 xenografts were inconspicuous. So considering the fold change in luminescence at endpoint, 134 cells were only marginally different from MCF7 cells, whereas the growth of some 44 xenografts differed by order of magnitude. So next, to analyze the growth of the different breast cancer cells in the context of the mouse ductal system, we stain whole mounted mammary glands with carminalum. So carminalum is a dye that reveals the milk ducts with their densely packed epithelial cells, as you can see here, the stereoscope microscope image, which reveals a dichotomously branched ductal system with a few side branches, which is characteristic of the young adult virgin mice that we used in this experiment. So 2 months after injection, the xenografted NST-MCF7 cells caused widespread dilation of the host ducts, with tumor cells growing evenly distributed through the ducts, but sparing the tip of the duct, really. So in contrast, the ILC, both ILC cell lines, homed to the ductal tips and gave rise to grape-like structures, pointing to a morphogenetic property, intrinsic morphogenetic property of ILC cells, resembling the terminal ductal lobular unit seen in the human breast. So next, we study a bit the histology of these models to further characterize them. And we nicely see histologies that we see in the clinical samples, the characteristic in situ single file invasive growth pattern, and the in situ, in parallel with in situ lobular carcinoma, and pointing to a more pleomorphic subtype on these cell lines. So also there are differences between the cell lines. The 134 xenografts had the K67 around 42, but it was highly heterogeneously expressed, and it was generally higher in the invasive than in the in situ components. And also 61% of the tumor cells here was ER positive and very heterogeneously expressed also in the mammary duct, the estrogen receptor. So while this was not the case for the SAM44, and also the estrogen receptor status of the xenografts resembled that of the clinical counterparts, the estrogen receptor expression. However, progesterone receptor in this setting was very low. So then we asked the very interesting question for us that is the hormone responsiveness, this model can be really induced by estrogen target, like to see progesterone receptor that is expressed. And this is the case. So stimulation of the host mice with estradiol increased the progesterone receptor index in both models to nearly 35%, indicating that the estrogen receptor progesterone receptor axis in this setting is very well preserved. So next, we study the metastatic potential of the ILC models, and both ILC cell lines were first detected at distant sites, here with the very sensitive ex vivo IVIS technique, only 5 months after injection, meaning that there is a long dormant phase in these models, in these lobular models. And the interesting thing is that the ILC cells spread more frequently in adrenal glands, GI tract, ovaries, brain, and peritoneal cavity, which is something that you expect from the information we have from the clinics. So finally, in these mice that xenografted with ILC cells, the radiance, so the signal at the endpoint in the metastatic organs increased dramatically, and it was really comparable with the primary tumors at 12 months after injection, where macrometastases appear. So having established that the intraductal microenvironment preserves the distinct biological characteristics of the ILCs versus the non-lobular ER-positive breast cancers, NSTs, we sought to determine the molecular underpinnings of these differences between ILC and NST carcinomas using a more relevant, patient-derived xenografts. So we took primary ILCs, treatment-naive samples from different patients, which we obtain and mechanically and enzymatically dissociate to single cells. We lentivirus transduced with luciferase GFP, and then we inject it intraductally. So in this way, several PDX were expanded by serial engraftment, and the pure tumor cells from 3 ILC and 3 NST are positive PDX were isolated by FACS. and RNA-seq was performed in that setting. So interestingly here, the principal component analysis separated the pure tumor cell samples by histological subtypes. That's pointing that there are tumor cell intrinsic differences in the global gene expression profiles that we see here in this preclinical model. Interestingly, the reactome pathway analysis of the differentially expressed genes revealed pathways, 7 pathways here that are enriched in ILCs, and consistent with the loss of adherin, we see cell junctions, hemidesmosome assembly, and cell communication genes that were among the top term genes. Now, surprisingly, given the fact that there are only tumoral cells here in this setting, with the FAC sorting that we perform, we observed that the extracellular matrix organization ranked as the most enriched gene signature. So furthermore, a gene set enrichment analysis identified gene sets for lobular versus ductal carcinoma, and adherin target genes significantly enriched in the ILC cells, validating the whole approach. So now to test if extracellular matrix remodeling signature, which we identified with this approach that I just described, is clinically relevant, we interrogated TCGA breast cancer datasets for ILCs and NSTs. So here you see the unsupervised hierarchical clustering based on the expression of 44 collagens and 195 glycoprotein-related genes derived from the matrisome signature, which mostly separated ILC from the NSTs, showing that the patient ILC are indeed enriched for ECM coding genes. So here on the right, you see the normalized counts where we notice that the elastin and 3 collagens were among the most highly enriched transcripts in ILCs. So this was very intriguing for us. So then we asked the question of whether ECM remodeling has really played a role in the genesis of ILCs that might be therapeutically exploited. So to address this question, we assessed the expression of enzymes which are responsible for cross-linking of collagens and elastin that was highly expressed in the ILCs. Because this family, they can be pharmacologically inhibited, at least preclinically. Here in this heat map, you see the normalized expression of the LOX family members and elastin. And as expected, ILC tumors show low adherin expression and high elastin expression. Now, the LOX family members, LOX, LOX2, LOX3, LOX4 were expressed at all comparable levels in ILC, and non-lobular ILC ER-positive breast cancers, but specifically here with LOXL1 transcript levels were different and 3-fold higher specifically in ILCs. So we focus our interest in LOXL1, which is it's a member of a family consisting of 5 paralogs, has a key role at maintaining extracellular matrix stability and integrity. And interestingly, this gene has been reported that has high copy number in another disease that also has adherin deficient tumors, is a diffuse gastric carcinoma. So now, in the light of these findings that the strong ECM signature is increased and LOXL1 expression in ILC, we examine LOXL1 transcript in actual patient samples with RNA, with RNA scope on the left and at the protein level on the right. So we found with both approaches, and especially you see here, interestingly, on the right with immunofluorescence, with the anti-estrogen receptor antibody in this invasive part of this invasive carcinoma sample that we tested, we saw a colocalization of LOXL1 with estrogen receptor expression. Thus, we concluded that LOXL1 can be expressed by lobular carcinoma cells, invasive lobular cells in patient samples. So now to test whether LOXL1 function is required for tumor growth and progression, and then we can use it as a therapeutic target. we made use of the natural compound beta-aminopropionitrile, BAPN. So this small molecule is a irreversible inhibitor of the active side of lysyl oxidase, the whole family. And the growth of ILC xenografts was statistically decreased. And also the metastasis, as we assessed here on the right, you can see in selected organs like lung, brain, and ovaries was also reduced. So now, to identify the changes, the molecular changes for LOX inhibition, we perform RNA-seq. And interestingly, we found that the principal component analysis separated the samples by the treatment, indicated that ILC tumors respond to the drugs. So now, interestingly, reactome pathway analysis or bioinformatic analysis of the differentially expressed genes revealed 7 biological pathways that are negatively affected by the BAPN treatment in the lobular tumors, and all these pathways are related to cell cycle progression. So now, however, the BAPN, the drug, inhibits the activity of all family members, and some of which are widely expressed. So we wanted to see if we can perform specific inhibition of LOXL1. So then we tested specifically LOXL1 with a genetic approach. So this down modulation with the shRNA of LOXL1 expression, and we performed different SH constructs, SH LOXL1 constructs, and the silencing efficiency was around 85%. So then we infected RFP luciferase expressing ILC cells with GFP scramble or GFP-SH LOXL1 expressing lentivirus, and following FAC sorting for GFP expression, the cells were engrafted intraductally. So here we assessed the engraft mammary glands at sacrifice with a fluorescent stereomicroscopic analysis, and that reveals that the ratio of the GFP-RFP signal emanating from the control glands was close to 1 and decreased by 1/3 in glands grafted with the GFP-SH LOXL1. So that indicating that the presence of SH LOXL1 bestows a proliferative disadvantage of these cells. So to summarize, we overcome the lack of the in vivo ILC models by generating robust ILC xenografts through intraductal engraftment. So these xenografts resemble the human ILC very faithfully. So we established some additional PDX ILCs with this approach. And when we took the global gene, and we analyzed by global gene expression the pure tumor cells from ILC or NST, and we profiled them, we found that extracellular matrix remodeling is a key tumor cell intrinsic ILC feature that connects the loss of Fic adherin with the production of ECM, as we saw also elastin and other collagens are produced by these cells intrinsically. So the inhibition of LOXL1 slows the tumor progression in this preclinical model that we tested. So to summarize overall about the models, so our aim now is, and general aim, I think, and we need a collaborative efforts for that, is to expand the current panel of PDX xenografts or GEMs. We really need this. We need to focus on the preclinical intervention using the PDX and the xenografts. We need to develop animal models to compare the primary versus metastatic ILC, or the classic ILC that we are missing, or the rare ILC triple negative tumors, for example. So we have to establish really some PDX that we are missing, or cell lines even from human ILC that have like wild type P53 and potentially an activating PIK3CA mutation. And with these models, we have to improve better and to study dormancy and metastasis with all this peculiar metastatic pattern that we see in ILCs. And we can potentially think about additional animal models that can help in this approach. But more overall, we really need a collaborative effort. So with this, I would like to acknowledge the people who contributed to these studies and express my gratitude to Professor Catherine Brisken here in EPFL for her enthusiastic support of the lobular preclinical studies that I'm performing in her lab. So our fantastic collaborators, all our generous funding sources, and but most importantly, I would like to acknowledge all our patients and their families here in Lausanne. Our patients not only inspire this preclinical work, but are at the center of our preclinical discovery process and all these efforts that we are doing. So thank you for listening, and I'm happy if you have any questions today. Thanks a lot.
Presentation. We have some questions from the audience. The first one, grape-like structures you see in ILC. Do you think that knockout of adherent in MCF7 will result in a more ILC-like morphology, or do you think it's more complicated?
Sorry, Dr. Patrick, just can you repeat the question, or is it, maybe I can read it, or because I couldn't hear well? Sorry, I cannot hear. Yes. Is there something wrong with my microphone, or?
No, you're good. I'm not sure what happened to Dr. Derksen, but you can read the questions on the right-hand side in the Q&A section.
Is it in the, okay, let's just a moment.
Microphone working now?
Yes, I can hear better.
Okay, yeah, there's something with my computer. Sorry about that. So the question was about the grape-like structures you see in ILC.
Yeah.
The knockout adherent in MCF7 will result in a more ILC-like morphology, or do you think it's more complicated than that?
Yes, so that's a great question. I think one of the key determinant of this grape-like structures can be the IC adhering loss. Because we also, I didn't present here, but we have it on the paper that we published recently, that we see, in contrast to the NST MCF7 cells, we see an intraepithelial growth also of these lobular cells. So we really see that they coexist with the mouse luminal cells, and they don't really fully replace and eliminate the mouse luminal cells. So they can just kind of mimic the pagetoid spread, so meaning that they are kind of more motile. I mean, they can move freely to inside the ducts, and at some point they establish themselves at the tip. But yeah, we don't know further mechanisms. But a driver of this phenotype for sure is the IC adhering loss. Yes.
Yeah, I think it's a very good point also to take into account. If you're injecting intraductally, you're physically restricted by the diameter of the duct, which is about 10 to 15 microns at the tip of the mouse duct, right? So by definition, you're going to end up with single cells in the ducts at the end, and the ILC cells will stuck in there. So that might be an explanation as well. I have another question.
Yeah, however, sorry to comment here, however, they make additional ducts, I can say, or TDL unit structures on the tip of, so they are kind of remake the TDL units by themselves. It's not only that they start there and they make a big bulb.
Okay, that's a very, very good point. Thanks for that. George, can you comment on the estrogen levels in the MIND model? And this is a question from the audience. Since PR is very inducible, is there room for adding exogenous estradiol? And do you know how that impacts on the progression time of your model?
So yes, that's a good question. Is really, yeah. So normally, yeah, we have a mass spectrometry in the lab that we are measuring the endogenous mouse levels, estradiol mouse levels. So these are low. Actually, these are mimic the postmenopausal women, and even lower, can be even lower. So, and at that levels, they, yeah, you see the progesterone receptor is not expressed, is inducible, yes, when we add the pellet. So, yes, I mean, considering that ILCs are very sensitive to aromatase inhibition, for example, yes, I think that there is room for adding exogenous levels in a very, very, very low amount comparing to what we normally do now. With the pellets, we put a level that is mimics the premenopausal women. Yes, and we are testing some conditions to adding on lower levels in order to kind of continuing having some estradiols that are not inhibited, you know, the growth of the ILCs finally, because they are so sensitive to aromatase inhibition.
Thank you. We have another question from the audience, and that is, does LOXL1 show any effect on tumor growth in IDC NST model or cells? And I had a similar question because one has also been identified as being an EMT-like effect, which might induce proliferation, right? Whereas it has also the cross-linking effect on collagen. So what do you think is important within ILC and in IDC?
Yeah, so we actually we try to test this in the NST cell lines, but we, you know, MCF7 cell line, for some really weird reason, the LOXL1 gene is over amplified. So there are many copies, but especially on MCF7. So for these reasons, we took PDX that we had here from fresh samples from the hospital that express estrogen receptor, and we hope that in this setting the LOXL1 was not amplified, and actually we include this in our study, and we didn't see the same effect. We didn't see inhibition on the primary growth of the PDX, not on the cell lines. So, but overall, it seems that IC adherine loss with the elastin that we see and the collagen overexpression, it seems that there is a reprogramming there in the lobular cells that they express some levels of genes and enzymes that you normally expect to be expressed by fibroblast. But in that case, intrinsically, also the lobular cells, they can facilitate what the fibroblast potentially can do in that setting. And I think it's really correlated with IC adherine loss.
Okay, so your point of the fibroblasts is a good one, because there's a question from the audience on what the balance is between LOXL1 expression by the tumor cells versus the carcinoma-associated fibroblasts. Do you know anything about that?
So, you know, in the primary samples that we tested with the RNA scope and the protein stainings, yeah, surprisingly, we didn't see a LOXL1 expression in cells other than the ones that express estrogen receptor in that setting. So probably, yeah, we didn't have a very enriched fibroblast enriched tumor samples there. So yeah, this is very interesting, and yeah, we would like to test, but we will need more clinical samples, so potentially a tumor microarray or something else to really test, or samples or lobular samples that are enriched with some fibroblasts, because there are also different type of fibroblasts. Not all they express LOXL1, I guess.
Thank you very much. There's a question from the audience on how representative the PDX models are for the ILC tumors from menopaused patients. And did you do any investigations on what the effect is of a hysterectomy in your mouse models? So
the PDX it depends. So in the PD adenosine mice, as we use as it is, like in that setting that the data I presented, we didn't put estradiol to grow. So these are not representative of so it's menopause. So the question is about post-menopause. It says menopause.
Yeah, so for menopause patients, I'm anticipating that the person in question asking if there is a different
so if
from
the
an
question,
point of view.
Yeah, so if the question is about post-menopause sample patients, so
yes,
the model we are using mimics these post-menopause. Yes, so we have studies that are ongoing, and we perform ovarectomy in order to see the effect on these lobular cells. So these are ongoing studies, but yes, I can tell already that there is an effect in ovarectomy on these mice where we eliminate the endogenous estradiol level. There is a massive effect on the proliferation of the cell lines in this setting. They can still grow, but they can be massively reduce their growth. Yeah.
Right, George, and a question from the audience. Given your late appearance of metastases in the model, do you do any investigations on whether you could find tumor cells?
So, we are currently looking on this because this is a unique opportunity to look about dormancy and overt metastasis. So, as I mentioned, so these are big experiments. They last up to a year. So the metastasis, it's a late event on this model, and but at that point is overt metastasis. So we are in the process of characterizing these overt metastases, and we are very intriguing about the fact that initially the first 5 months, 4 to 6 months, were very, very long time after engraftment. We don't really see a massive. We don't see metastasis with these sensitive techniques. So it seems that there are very little dormant cells at that stage. So potentially there is a window in the following months up to 9 months or a year that a small window, I would say, because then at some point these cells, surprisingly, they metastasize more than other models that we have tested in the past in our hands for NST models on MCF7, that they stay dormant for a long time. This is something we would like to explore further, yes.
Right. So I was also thinking about the long time it takes to establish tumors. So have you checked the genetic status of the cells that grow out after 7, 8, 9 months versus the ones you inject, whether there are there's an accumulation maybe of pro-oncogenic hits?
Yeah, so yeah, we don't have data on this, but this is something that we want to analyze. Yes, I don't know the.
Yeah, difficult question. Thank you very much, George, for this beautiful presentation, and thanks to the audience for all the great questions.
Thank you very much.
All right. So we will move on to the final speaker of this session, which is Jeji An. Jeji is doing or did, I'm not quite sure, her PhD at John Egan's lab at the Department of Molecular Genetics at the University of Toronto, studying breast cancer using mouse models. And the focus of her research has been mainly in lobular breast cancer and the relevant genetic events associated with lobular tumors. So the title of today is Refining the ILC Mouse Model, Exploring the Role of 16q Hemizygosity in Breast Cancer. So Magee, if you can share your screen, and can you stop sharing your screen, George?
Oh, great. Oh, I can't see it. There we go. Okay. Can you see my slides?
Yes, we can see it.
Okay.
Thank you.
Okay, perfect. Yeah, so thank you so much for the introduction and for this opportunity to speak. So today I'll be presenting on an ILC mouse model that we've developed, and also sharing some of our work on investigating the loss of the hemizygous loss of chromosome 16q in breast cancer and including ILC. So ILC has been molecularly characterized and categorized into different subtypes and based on their transcriptional profile by different labs. And of course, I'm sure everyone has been talking about CDH1, which codes for E-cadherin, but it is the most frequently mutated gene in ILC. And most of these mutations are truncation mutations, shown in black here. And the second most common mutation is hotspot mutations, shown in orange in this figure in PIK3CA, which codes for the catalytic subunit of PI3 kinase. And these two mutations often co-occur in patients. So based on this, we've developed a mouse model using these two mutations. And this work was published in 2018. And I just want to acknowledge some of the key people who brought this paper together. So there was Jess Adams, who was a research associate in the Egan lab, and she generated most of the mouse cohort. And there was also Daniel Holland, who was a postdoc in the Peru lab, and he processed the transcriptional data. And our main collaborators were Chuck Peru and Cindy Guidos, and my supervisor, Siobhán again. So in this paper, we showed that homozygous loss of CDH1 and PIK3CA hotspot mutation of H1047R cooperate to promote mammary tumor development in the mouse. So this is this brown line here, and the mammary tumor-free survival of these mice were significantly decreased compared to PIK3CA H1047R on its own, which is the orange line here. And so PIK3CA H1047R tumors generally were mostly serous tubular carcinoma histotype. So these tumors had spindle cells and very fibrous regions. But with complete loss of CDH1, the histology shifted towards serous tumors, or STs. And these also have spindle cells, but instead of being very fibrous, they had more invasive borders. And so the CDH1 PIK3CA mouse model shared many features with human ILC, such as the single file lines, the targetoid patterns, abundance of collagen, which is shown in blue here. I also confirmed that E-cadherin expression was lost. These tumors were also positive for estrogen receptor, and the tumor cells had formed these microvilli, which is another unique feature of ILC. So once we established that the mouse model had many shared histological characteristics, we wanted to see if the tumors were molecularly similar to human disease. And sorry, there's just someone at the door. And also we wanted to determine which ILC subtype that our model might fall into. So in order to do that, we compared the transcriptional profile of our mouse model to human data. So this is work done by Daniel Holland. So our model clusters the immune-related ILC subtype. So as the name suggests, the immune-related subtype has a high immune signature. And here I'm just showing you some of the immune cell expression in the human tumors and our mouse tumors. So high expression is shown in red, the lower expression in green. The first column is showing all the luminal A ILC human tumors. And then the second column is showing the luminal A immune-related ILC tumors, human tumors extracted from this first column. And then right beside it is our mouse model, which you can see is very similar. And then beside that is all the luminal A IDC human tumors, and lastly, other mouse mammary tumor models. Just wanted to show that our model is very closely related to the IR-ILC model. The other thing I wanted to point out was that there was a high expression of the CTLA4 and CD1 signaling pathways in both the human ILC tumors and our mouse tumors. which suggested to us that immune checkpoint therapy may be effective in ILC patients and that we might be able to test this in our mice. All in all, we wanted to better understand the immune landscape. So we ended up running a Seitoff experiment. So this is work done by the Guidos lab, specifically by Magee and Anthony Zhao. And Seitoff, or mass cytometry, is a combination of flow cytometry and mass spec. So we use antibodies tagged with metal ions in order to label the cells. And the data is presented as phenographs, which is a 2D representation of the multidimensional data. So here we're looking at a phenograph of T cells only, and the different colors represent different T cell populations. In the control sample, which were normal mouse mammary glands, and then the tumor samples down here. So I just want to bring to attention a few populations. In the top left corner here, there's this cobalt blue and navy blue population that is pretty abundant in the control, but has decreased in the tumor. And there's also this very light blue population that's very abundant in the tumor, which has decreased in, or not decreased, but is not very present in the control. And lastly, this green population here at the bottom is more abundant in the tumor versus control. So let's take a look at what these populations were. So here the red color actually represents high expression of the antigen here, which is CD8 beta, and it's a marker for cytotoxic T cells. So these two populations in the control sample were cytotoxic T cells, and the light blue in the tumor were also cytotoxic T cells, but you can see that there's a shift in the kinds of other antigens that they are expressing, and therefore it clustered differently in the control and tumor. And then the green population I mentioned is the FOXP3 positive cells. And so this is a marker for regulatory T cells. So there was an increase in regulatory T cells in the tumor compared to control samples. And both of these populations highly express PD-1, which is an inhibitory cell immune checkpoint marker. And all of this suggests to us that there is an immune exhaustive phenotype in the mouse tumors. Okay. The Seitoff data also revealed a population of cells that was much more abundant in the tumor compared to the control, this navy population here. So here we're actually looking at myeloid populations in the control and tumor samples. And again, the colors represent different clusters of cells. And so this population were identified as tumor-associated macrophages by the marker CD11C. So here again, we're looking at the expression level of CD11C. That's what the colors represent. And these cells had high expression of MHC2, which suggested that these tumor-associated macrophages were active. Okay, so just to summarize the first part of my presentation, I showed you that our mouse model of ILC had very similar histological and molecular features to human IR ILC. And that this model can be used as a platform for therapy development. So the next part of this project is actually being carried out by Adil, who is another PhD student in the lab. And she crossed these mice with preconditional luciferase mouse line, which will help monitor the tumors using bioluminescence imaging. And so the mice will be used to conduct preclinical trials of different combination of therapies. So like the immune checkpoint therapy and hormone therapy and small molecule inhibitors. Okay, so moving forward, we wanted to further refine our ILC model and also understand other very common genetic events in ILC as well as breast cancer in general. So one of these common events is actually hemizygous loss of the long arm of chromosome 16, 16q here. So this region encompasses CDH1, which again codes for that popular E-cadherin protein in ILC patients. And most ILC patients actually lose E-cadherin expression through focal mutation one copy of CDH1 and losing the other copy through 16q hemizygosity. Actually, many breast tumors lose the whole arm of chromosome 16. So here we're looking at 10 genes across 16q, and most of these have hemizygous loss, shown in this light blue color. Okay, so the most commonly deleted region on chromosome 16q is 16q22.1. And this, again, carries candidate tumor suppressor gene, CDH1, as well as another gene we're interested in, CBFB. So both of these genes are frequently mutated in breast cancer, and they both experience homozygous loss in breast cancer through heterozygous deletion of one copy, usually due to losing 16q, and loss of function mutation in the second copy. So that suggests to us that they might be acting as recessive tumor suppressor genes. And I've already shown you with CDH1 that it does act as a recessive tumor suppressor gene in cooperation with mutant PIK3CA. And so here, the tumors with a focal deletion and heterozygous loss of CDH1 are mainly ILC tumors being represented. And interestingly, the CDH1 mutations do not co-occur with CBFB mutations. So we were interested in seeing what the role of CBFB was in breast cancer, and we also wanted to test whether CBFB may be acting as a haploinsufficient tumor suppressor in ILC models or ILC patients. So CBFB codes for a protein called core binding factor beta, and it's a binding partner of the RUNX transcription factor family. And CBFB and RUNX proteins affect the expression of different genes, and they also impact mammary epithelial cell differentiation. And interestingly, RUNX1 is the third most mutated gene in ILC, which suggests to us that the RUNX1 pathway may be important in ILC development. Just to summarize, in ILC patients, they have a hemizygous loss of CBFB through 16q loss, and then some patients have mutated RUNX1. Okay, so much like with CDH1, I made heterozygous and homozygous loss of CBFB with or without mutant PIK3CA mouse models. And so what I found was that homozygous loss of CBFB cooperated with active PIK3CA as a recessive tumor suppressor gene. So this is this dark blue line here, and it was significantly faster than the mutant PIK3CA on its own, which is this orange line here. With heterozygous loss of CBFB, it cooperated with PIK3CA in order to decrease tumor latency, but it didn't impact penetrance. So it wasn't, the memory tumor-free survival curve wasn't significantly different, but it did significantly impact tumor latency in these mice. So as I mentioned, CDH1 and CBFB is usually lost through 16q hemizygosity rather than focal mutation. So we wanted to study losing 16q in the context of breast cancer. So we worked with Laura Nutter from the Center for Phenogenomics, or TCP, in order to model 16q hemizygosity in mice. So the cool thing about human chromosome 16 is that or 16q is that there's a syntenic region on mouse chromosome 8. So we were able to flank this region with LOXP sites in order to create a preconditional 16q deletion model. So losing this 16q syntenic region on mouse chromosome 8 was sufficient to induce mammary tumors in mice. So this is this orange line here, and it was significant compared to Cre-negative and MNTB Cre controls. And the tumors from these mice were mostly from the spindle family, these pink families here, which is the same histology family as the ILC model that I just showed you before. And this is what these tumors look like. They both have spindly cells. It's just that STs have more of an invasive border than the SCT histology. Okay, so we sent these tumor samples to the sequencing facility at SickKits, TCAG, and they did whole exome sequencing for us. And we were able to show that there was hemizygous loss of 16q, the synthetic region on mouse chromosome 8. And all of these tumors had amplification of MET. And so a subset of breast cancers has 16q loss and MET gain and amplification. So 16q loss here is represented by VPS35, CBFB, CDH1, and GAS8 hemizygous loss. So VPS35 and GAS8 were the N genes on the region of mouse chromosome 8 that was flanked by LOXP sites in our model. So yeah, these two events do occur in some breast cancers, but MET amplification and gain isn't very common in ILC. So we want to conduct a sleeping beauty screen with the 16q hemizygous mouse in order to find other cooperating events with 16q loss. So in particular, we're interested in genes that might be on the other chromosome arm losses and gains that co-occur with 16q. So for example, 16q loss is often occurs with gain of 1q. To this end, we are we have bred mice with a Sleeping Beauty machinery and 16q loss. So for those of you who may not know Sleeping Beauty, there's a transposase in the mouse ROSA 26 locus, and once it's transcribed, it cuts out and inserts transposons all over the genome randomly, and this can result in enhancement or disruption of gene expression. And so we anticipate that certain events will be selected for in the resulting tumors, and that we can map the transposon insertion sites and then identify the mutated genes that may be cooperating with 16q loss. Okay, so in summary, I showed you that we developed a novel mouse model for immune-related ILC, and that this model can be used as a platform for drug discovery. I also showed you that CDH1 and CBFB both act as recessive tumor suppressor genes in cooperation with mutant PI3 kinase, and also that single loss copy of CBFB affects latency of PIK3CA memory tumors. And lastly, I showed you that 16q hemizygosity is sufficient to induce memory tumors in mice, which suggests to us that they may be playing a role in ILC because so many ILC tumors have this genetic event. So moving forward, we hope to identify differences between the ILC mouse models with or without 16q hemizygosity in order to try to help guide therapy development. So to do this, we are working with Leigh from Spark Biocenter at SickKits to run a protein, to perform protein analysis, and we'll also be carrying out transcriptional profiling to identify these differences. And as I mentioned, we're also doing a Sleeping Beauty screen in order to identify any cooperating oncogenic networks beyond amplification of MET that we saw in our sequencing data. So these mice have been generated, and we're currently processing these samples. And the last thing we want to do is to explore synthetic lethality. So synthetic lethality is the concept that two mutations do not compromise viability individually, but when they're combined, they are lethal. So we want to perform an RNAi or CRISPR knockout screen with the 16q hemizygous mouse model in order to identify synthetic lethal partners of 16q loss, which will then become candidates for novel therapeutic as novel therapeutic targets. Okay, so with that, thank you so much for listening to my presentation. I would like to thank my supervisor, Siobhán, as well as current and past lab members. In particular, I want to mention Jess, who generated the IR-ILC model, Caitlin and Adil, who are current PhD students that are also working on different ILC projects in the lab. My supervisory committee, our collaborators, particularly Cindy and Mickey and Anthony from her lab, as well as Chuck and Daniel from the Peru lab, who did all the transcriptional profiling. Susan Doan, who is a breast cancer pathologist at Princess Margaret Hospital, she shared her expertise with us in breast cancer pathology. And lastly, the helpful facilities at SickKids and UHN. So Laurel Nutter, who made the 16q model, and then Leanne, who did the protein analysis. Sorry, I also want to mention our funding partners. Yes. So thank you, and I can take your questions now.
All right, thank you, Magee. This was an impressive amount and a high quality of work you were showing. We have a question from a patient advocate. This is the first time I've heard of Sleeping Beauty. Is this a process similar to CRISPR? So maybe you can explain a little what the differences are between the CRISPR-based techniques and the Sleeping Beauty you're using in your mouse models.
Right, okay. So I guess Sleeping Beauty is taking advantage of naturally, well, there are transposons that are found naturally, but we put in, we have mice with transposons that have mice that have been engineered to have transposons, and they are paired with another mouse that has a protein called transposase, and the transposase cuts out the transposons and inserts these random sequences that can either promote or disrupt genes. Whereas CRISPR would, I guess, like CRISPR has a guide RNA that helps it to target specific genes and locations in the genome. So if you're doing a CRISPR screen, you would have like a library of different guide RNAs and then target a different set of genes. Whereas I guess it's a little bit more random with Sleeping Beauty, where the transposon can jump around anywhere in the genome, and we end up seeing the ones that give an advantage to the tumor cell in the final set of genes that we get. I don't know if that was a very good explanation, but
it's a complex method. You did a very good job. I think if I can add anything to this, we consider a transposon type of a virus that integrates into your DNA and surrounding it, and then you can identify the genes that are surrounding it. And with CRISPR, you use an enzyme to cut the DNA into a little piece. So that's a different technique. Yeah. All right, thank you very much, Magee. Another question from the audience. Do CBFP mutations co-occur with PIK3CA mutations in ILC patients? Have you looked at it?
Yeah, so CBFP mutations do co-occur with PIK3CA. I'm not sure if they specifically co-occur in ILC patients. That may happen, but I'm not 100% sure that it does. I know it does happen in breast cancer in general. Yeah.
Okay, thank you. There's a talk from the audience, or there's a question from the audience, sorry, asking if you see lobular carcinoma in situ, so Elbcc in mice with homozygous loss of 16q.
In situ. Yeah, so actually 16q loss is a very early event, and it has been seen in situ in ILC and other breast cancer subtypes. Was that the question?
I think so, yeah. I have a question about your immune response and the idea to use this as a model for immune inhibitors. So we heard yesterday from the Gelato trial that the responses are mainly in the patients that are pleomorphic or even triple negative ILC, whereas the more classical ILC do not respond to immunotherapy. And I'm asking the question also because in the previous mouse model done by the Jokkers group, in which they mutated PI3 kinase, they claimed that this was a classical model, and your model seems to be more immune-driven. So do you think that your model is more pleomorphic than the ILCs from the NKI?
I'm not sure. Maybe it is more pleomorphic. I definitely see some pleomorphic characteristics in terms of the when I look at some of the nuclei, but we haven't looked too deeply into characterizing it in that way. It is very invasive. And when we did talk with the pathologist, she did mention that there were some potentially pleomorphic features in our model, so that might very well be possible. But yeah.
Thank you. There's a talk from the audience. There's a question from the audience. I keep making the same mistake. In the Sleeping Beauty screen you're planning, are you planning to include a group that has also lost the other copy of cadherin to mimic the truncating mutation seen in ILT?
Well, we haven't actually thought of that far ahead with the Sleeping Beauty screen, but we've definitely have models where we have deleted the other copy of CDH1 and have the hemizygous 16q with PIK3CA, and we see very similar, we get very similar tumors as the model where we've deleted CDH1 just using focal mutations. But we currently don't have plans to do CDH1 and 16q and Sleeping Beauty all together, but that is very interesting.
Okay, just out of curiosity, I saw you switched from the WOCRE models to an MTV-driven model. Why is that? Because the outcome of inducing mutations in these models is quite different from a phenotypical point of view.
Part of it was for ease of the experiment. So with WOCRE, as you probably know, you have to breed them in order to activate the promoter, whereas with MNTV, that's you don't have to breed them, and so it was easier to, like, less labor intensive to generate these mice and to turn on CRE. And I think we were also interested to see if perhaps we'll get something different with the MNTV CRE driver, just because we're targeting different cells that would be expressing CRE. So I think those were the two reasons why we switched over.
Okay, thank you very much. I think one more question from a patient advocate. The question is loss of 16q versus mutation, is that an important difference?
Do you mean like mutation as in CDH1? Because 16q, like the whole, sorry.
I think the question stems from being unfamiliar with terminology of loss of a whole chromosome arm versus mutation on a given gene. Maybe you can comment on that.
Yeah, of course. So loss of a whole chromosome arm is different from having a focal mutation in a gene, because you're actually losing one copy of whole array of different genes with losing the whole arm of 16q. So what we are thinking is that there might be like compound haploinsufficient tumor suppressor effect, where losing one copy of all these genes might have a compound effect and maybe cooperating together to drive tumors. Whereas like losing just one, like having a mutation in one gene may not have as a mass effect. It's also like modeling a focal mutation and studying a focal mutation is also much easier. It's hard to tell which genes are actually important on chromosome 16. So that's something else we're interested in parsing out. So yeah, it is different. We're just not sure exactly how important it will be in moving forward and in therapy and disease progression. Yeah.
Okay, thank you very much, Yi. I would like to thank all speakers for brilliant talks and the audience for a good question session. I think we'll wrap it up here. And if I'm not hearing anything from Steffi or Otto, I think we should reconvene at 12:30 Eastern Standard Time for the next session. So thank you very much.